In the United States, 1 to 6 per 1,000 newborns demonstrate some form of measurable hearing loss. Auditory input is the mandatory physiological prerequisite for linguistic, cognitive, and social development. Any impairment during the first 18 months of life deprives the brain of the acoustic stimulation necessary for language. This window of sensory deprivation triggers an irreversible developmental cascade. Universal Newborn Hearing Screening, or UNHS, acts as the primary clinical preventative measure to identify these impairments at birth. Successfully executing the UNHS protocol requires clinicians to reverse engineer the ear's mechanical and neural pathways to find exactly where a signal fails. To reach the brain, an auditory signal must transition through 3 distinct mediums: acoustic waves in the air, mechanical vibration through bone and fluid, and electrical action potentials in the nerves. The external ear initiates this process. The cartilaginous framework of the pinna and the external meatus are shaped to capture and funnel ambient acoustic waves inward. While a full-term canal is firm and patent, the canal in a neonate born before 34 weeks is often collapsed, which can temporarily obstruct the acoustic pathway. Incoming waves strike the tympanic membrane, causing the ossicular chain, the malleus, incus, and stapes, to vibrate. The chain serves as an impedance matcher, driving a physical wave into the fluid at the oval window and translating air pressure into fluid motion. The outer and middle ear exist to convert acoustic energy into fluid motion without signal degradation. Once the signal reaches the inner ear, it enters the cochlea and the organ of Corti. As the fluid wave moves from the base of the cochlea to the apex, it displaces the basilar membrane. This displacement forces receptor hair cells to bend against the overlying tectorial membrane. This mechanical bending triggers transduction, the conversion of physical displacement into electrical action potentials. These electrical impulses exit the temporal bone via the fibers of the 8th cranial, or vestibulocochlear nerve. This process allows the auditory system to convert external physics into the electrical signals required by the brainstem and cortex. Conductive hearing loss occurs when a mechanical failure in the outer or middle ear disrupts the vibration sequence. Anatomical blockages like oral atresia or middle ear effusion stop the ossicular chain from vibrating, preventing sound from reaching the inner ear. External anatomical markers, such as pre-auricular tags or pits, often correlate with malformations occurring deeper within the ear structure. In these cases, the inner neural hardware, the cochlea and the auditory nerve, remains perfectly intact. Because the sensory nerve endings are undamaged, clearing the mechanical blockage or bypassing it typically restores normal hearing. Sensory neural hearing loss results from a failure of inner ear transduction or dysfunction of the auditory nerve. In conditions like Scheibe dysplasia, hair cells are missing or collapsed. When the fluid wave arrives, there is no mechanical action to trigger an electrical signal. Without transduction, the 8th cranial nerve remains silent, regardless of fluid motion. These structural failures are often genetic, tied to autosomal recessive inheritance, trisomy 21, or the connexin 26 marker. Because sensory neural loss involves the destruction of the biological transducer, interventions must bypass the physical anatomy entirely. The UNHS protocol uses a 2-tiered physiological filter to isolate exactly where the auditory signal stops. The first tier is the Otoacoustic Emissions, or OAE, test. A sensitive microphone in the ear canal measures low-intensity echoes produced by healthy cochlear outer hair cells. A passing OAE result confirms that the mechanical pathway up to the inner ear is functioning. If an infant does not pass the OAE, clinicians use the Auditory Brainstem Response, or ABR, test to measure the actual neural transmission. Electrodes record the sequence of waves produced by the auditory nerve. Clinicians specifically analyze the latency and presence of the wave V peak. A delayed or absent wave V confirms a sensory neural transmission failure, indicating that the signal is failing to reach the brainstem. Combined, the OAE and ABR tests provide a map that distinguishes a mechanical defect from a neural defect. This data drives the early hearing detection and intervention timeline: screening by 1 month, diagnosis by 3, and intervention by 6 months. The deadline is dictated by neuroplasticity. To build the brain's architecture for language, the auditory cortex must receive signals in this period. For permanent conductive losses, clinicians use external amplification to force a mechanical signal through the blocked ear structures. In the neonatal auditory system, delay is costly. Missing these windows allows the brain's architecture to wire itself for silence. For severe sensory neural loss, clinicians use cochlear implants to restore the signal pathway. The implant bypasses the damaged organ of Corti entirely. An electrode array delivers electrical signals directly to the spiral ganglion cells of the auditory nerve. An external processor converts acoustic sound into digital electrical signals, replacing the biological transducer. By accurately mapping and treating specific anatomical failures, early clinical intervention provides the acoustic signals a developing brain needs to thrive.